Welcome back, fellow scientists. So we are, just to give you kind of a frame of reference, we are in the thick of talking about the central dogma of biology, transcription and translation. Mr. Anderson from Bozeman Biology kind of gave us an overview 2 videos ago. Last video, I went in depth on transcription. This video, we're going to go in depth on translation. So I am going to go to the appropriate notebook page. I'm not going to say blank because that would be a lie because I already wrote stuff down. So you take out a blank notebook page and just to kind of summarize again, to try to give you a frame of reference, we're talking about the central dogma of biology, which is the fact that we have DNA, which is the master recipe, the master blueprint that's kept safe in the nucleus. Right? And that, through the process of transcription, that information is used to make RNA. So that's kind of the blueprint copy that goes out to the job site. That's the index card that goes onto the counter when you're baking something. That's the nucleic acid that goes into the cytoplasm that can be used, that can be degraded, and we don't really care because we can go back and we can make new stuff, right? We don't care if anything bad happens to it because we always have this master copy back in the nucleus, right? So that's transcription. And then today we're talking about translation. How do we take that RNA and what job does it do? How do we use those blueprints to make the building? How do we use that recipe to make the pizza? How do we use that nucleic acid to make the protein? And then of course proteins, we've already talked about, are the workers. If anything needs to be done, there's a protein that does it, and your proteins give you in part your characteristics. So your hair color, your eye color, your height, your weight, your all sorts of stuff, propensity for heart disease, propensity for cancer, is determined in part by your proteins. So your proteins make up you. So if you recall, that's the central dogma of biology. That's what we're driving at. That's the main thing. So let's go ahead and write down a definition for translation. So like I've already said, we're taking the information in RNA and we're putting that to work. We're using that information to make protein. So go ahead and write that down. I'll wait. You can pause the video if you need to. Perfect. Okay, good. Sorry, I drifted off to sleep there. All right. So what we're going to do now, before we go into the actual process of talking about translation, is we're going to look at the 3 different types of RNA. So we mentioned those briefly when we were talking about transcription, the difference between DNA and RNA. So now I want to go a little bit more in depth. So mRNA, we could kind of picture it like this. This line is the sugar phosphate backbone. And then we have bases, obviously, extending out of the sugar phosphate backbone. Right? mRNA is still made out of nucleotides. We already talked about it. mRNA just has an extra oxygen in its nucleotide. DNA is missing an oxygen, deoxyribonucleic acid. So it's the same kind of thing. mRNA is single-stranded. It's not double-stranded, so the bases don't pair up always or yet, as we'll see in the process. And one kind of cool thing about mRNA is that it's separated into groups of 3, or we like to separate. It's not physically separated, like the bases are evenly spaced in reality. But we like to think about it as being separated into groups of 3 bases, and those groups of 3 bases are called codons. Right? So here, AUG. So this is one codon. Right? So really quickly, quiz question, just to make sure you understand, how many codons are in this piece of mRNA right here? 5. Perfect. Because we have 1 right here, and then this group of 3 is 2, this group of 3 is 3, this group of 3 is 4, and then this group of 3 is 5. Good. All right. You guys got codon. So that's mRNA. Next one that we want to talk about is rRNA. Okay, so rRNA obviously stands for ribosomal RNA, so it has something to do with the ribosome. So I drew the ribosome here in black. If you want to use a different color, that might make more sense in your notes. So back in chapter 7, when we talked about cells and cell organelles, we only drew a ribosome as a small, tiny little dot. Right, Mr. Leo, were you lying to us? No, not really. I was simplifying it so that you wouldn't be overwhelmed by concepts. But now that you have the idea that a ribosome is in the cytoplasm or on the rough endoplasmic reticulum and it helps to make protein, now we can go even deeper and we can say actually a ribosome is made of 2 subunits. There's a small subunit and there's a large subunit. And it just so happens that both subunits have rRNA associated with them. Right, so a ribosome is part protein, part nucleic acid. It's kind of a hybrid of the 2. And so these pieces of rRNA, they help the tRNA to combine with the mRNA. Oh, okay, Mr. Leo, I get mRNA, fine, rRNA, that's a thing, whatever, but what in the world? tRNA, come on, help me out. Okay, you got it. So here is tRNA. Okay, so tRNA as stands for transfer RNA. Well, Mr. Leo, what are we transferring? Well, we're transferring the information from mRNA to a protein. Okay, now you got to remember all the way back to chapter 2, what are the building blocks of proteins? Yes, amino acids, which are right here. Okay, so check this out. This is the grand scheme. This is the process of translation. We're going to have our anticodon on our tRNA, our unpaired bases on this kind of loop-de-loop thing down at the bottom. We're going to have our anticodon. It's going to pair, UAC, right? It's going to pair with our codon, AUG. Complementary base pairing rules. Remember, mRNA, or RNA rather, doesn't have a T, it has a U. So A pairs with U, U pairs with A, and then of course G still pairs with C. All right, so this anticodon is going to pair with that codon, and there's an amino acid at the end. And this amino acid is specific for this anticodon, right? So we have 20 different amino acids. We have, 64 different anticodons. We'll get to that later. But I have 64 different anticodons, so there has to be at least 20, possibly 64 different tRNA molecules, right? And you can notice this is where I said that most of the times RNA is single-stranded, sometimes it's double-stranded. tRNA is an example of where it's sometimes double-stranded. And even in this molecule, you can see that some of the bases, right here, right here, right here, right here, some of those bases complementary pair, some of them match up. But some of them, they have these little loop-to-loop things right here, where the bases don't pair, they're not complementary, right? And this loop-to-loop thing right down here, this is the most important one. That's the anticodon that's going to match up with the codon that's going to specify which amino acid needs to go in the string that's going to make our protein. Okay, so that's the basics of the different types of mRNA. So now we're going to go through the process of translation, and we're going to use our before, during, and after motif, just like we did previously with DNA replication and with transcription. So go ahead and write down before. And before, it's really easy. All we have is just this little piece of mRNA, right? You'll notice that I omitted the letters. They're still there. These little lines are bases. But just for the sake of clarity, it would be really distracting if the letters were there. So just trust me, they're there. They represent bases. And our base pairing rules, still apply. They're still grouped into codons. So that's before. That's all you have to write for before. And then during, we're going to start with, obviously, our mRNA. And then we're going to have our ribosome. So the small subunit of the ribosome comes in and attaches, right? And then the large subunit of the ribosome comes in and attaches. So you can go ahead and draw that. That's what your first during picture should look like. Okay, so then the ribosome, you'll remember the rRNA helps the tRNA combine with the mRNA. So here's our little tRNA. It's not drawn to scale, unfortunately, but you can kind of see this base matches up with this base, this matches up with this base, this matches up with this base. So our anticodon on tRNA matches up with our codon on mRNA. And so this tRNA is going to kind of float in and is going to match up, and the ribosome is going to help it to do that. And then we're going to have another tRNA that's going to kind of float in and match up, right? This matches with this, this with this, this with this. And then this is the key part. This is the main thing that the ribosome does. The ribosome is going to take this little amino acid, if I can select it. Nope, come on. If I can select it, move. There we go. It's going to take this amino acid right here, and it's going to catalyze this reaction. It's going to transfer, that's why it's called tRNA, transfer RNA. It's going to transfer this amino acid from this molecule to this molecule, right? Once that happens, then our ribosome is going to move down. This tRNA is going to leave. All right, so maybe, I don't know how you draw that, whether you do two pictures or whatnot. It's kind of hard. You guys can figure it out. You're smarticles. All right, so now we have this tRNA that's out of there, right? And then we have this ribosome that's kind of moved all the way down. So then we have another, oops, come back here, little amino acid. So then we have another tRNA with an amino acid that's going to go right here. Okay, so that's to that, that's to that. So that base pair attaches to that, that attaches to that, that attaches to that right there. And then our RNA is going to take these two and is going to transfer them. Right, our ribosome is going to take these two from this tRNA and is going to attach them to this tRNA, and then our ribosome is going to move on down the strand, and that's going to go away, and then we're going to have another one float in, and then it's going to attach, and so on and so forth ad nauseum until we get to the end, the after party, right, where we have just our regular mRNA again. Okay, there was a release factor that came in and it kind of like blew the ribosome apart, and that's beyond the scope of this course. We'll talk about that in AP biology, but it's a thing. So that's how the ribosome blows apart, right? And then we have all these empty tRNAs that have given their amino acids to the string right here. All of these anticodons have matched up with these codons, and it's great and it's amazing. And then these are going to go off and be charged. They're going to be recharged. The amino acids are going to go back on here. Different amino acids, not these amino acids, but different amino acids from the food that you eat, right? You eat protein, you break it down in amino acids. Amino acids get attached to tRNA. tRNA attaches to mRNA. Ribosomes make protein, and then you make your own protein. So that's why you need to eat protein, so that you can have the building blocks to make your own protein that you need. Anyways, I digress. So there's another enzyme that charges these. Again, that's beyond the scope of this course. We'll talk about that in AP biology. But suffice it to say, we have our mRNA, which can be used again, right? Another ribosome or the same ribosome can come in and attach, small subunit, large subunit, right? And oops, we left a line. and then we can have new charged tRNA come in, and the whole process can start over again. But this is the main thing right here. This protein, that is the goal. That is the whole reason why we have DNA. That's the whole reason why we have RNA. That makes you. That protein, that string of amino acids. Say, Mr. Leo, that doesn't look very impressive. I know, because it's very simple. It's very simplified. This is just like 1, 2, 3, 4, 5, 6, 7, just 7 amino acids. That's not even technically a protein. That's a polypeptide, right? So proteins can be up to 150 amino acids, 450 amino acids, thousands of amino acids. The proteins that make up your muscles are thousands and thousands and thousands of amino acids long, right? So this is just a really simplified version, but that's the end product. That's why we have DNA. That's the process of transcription translation. That's one of the tools that God uses to make you, which is simply incredible, and we humbly stand in awe of his creation and rejoice in the complexity of it. So this has been Mr. Leo. Today we talked about translation in depth. Thanks so much for watching, and I will see you on the flip side.